Assalamu alaikum. You are watching Microbiology by Nouman. In this video, we will be talking about the most common staining procedure that is used in microbiology, known as gram staining. This gram staining technique was developed by Christian Gram in 1884. And on the basis of gram staining, we categorize bacteria into two groups. The first group is known as gram positive bacteria, and the second group is known as gram negative bacteria. On the basis of bacterial cell wall thickness, these two groups are made. We know that bacterial cell wall is made up of peptidoglycan or mucopeptide or murein. We can use any of these words, peptide, mucopeptide or murein. This bacterial cell wall, all those bacteria that are having a thick cell wall or their cell wall is made up of thick peptidoglycan, they are termed as gram positive bacteria. Bacteria containing thick peptidoglycan cell wall. Whereas all those bacteria that are having a thin cell wall, they are termed as gram negative bacteria. Gram positive bacteria, after this gram staining technique, they appear as purple in color. Whereas gram negative bacteria, after this gram staining technique, they appear as pink or red in color. But the question is how this cell wall thickness is determining whether the bacteria will appear as purple or pink or red in color. First, we will draw the structure of their gram positive and gram negative bacteria, and we will understand how this staining procedure works. If we look at the cell wall and the cellular structure of gram positive bacteria, outside this cytoplasm, cell membrane is present, right? Which is made up of lipids and proteins. This is cell membrane. Outside this cell membrane, a cell wall which is made up of peptidoglycan is present. There is a thick peptidoglycan layer present in case of gram positive bacteria. This peptidoglycan is basically made up of two subunits which is known as N-acetyl muramic acid and N-acetyl glucosamine, NAM and NAG, right? If we look at the cell wall of gram negative bacteria, outside the cytoplasm, we see a cell membrane is present, which is again made up of lipids and protein. Outside this cell membrane, a very thin peptidoglycan layer is present. This is cell wall, right? Which is made up of peptidoglycan. Outside this cell wall, another structure is present which is known as outer membrane. Now we are looking for how gram positive bacteria and gram negative bacteria are stained during this gram staining procedure. There are 4 steps, and we are looking for how gram positive rod and a gram positive cocci and gram negative rod and a gram negative cocci changes color during these 4 steps of gram staining. The first step of gram staining is we apply a stain known as crystal violet. This crystal violet will penetrate inside the cell wall of both gram positive bacteria as well as gram negative bacteria and cause both of these bacteria will be colored as purple. In the second step, we will apply a solution known as Graham's iodine. It is a yellow color solution that will penetrate inside bacterial cell wall and make a crystal violet iodine complex. It will bind with this crystal violet and make a complex. This is an insoluble complex. This Graham's iodine is acting as a moderant. Also, it will kill bacteria as well. So bacteria will be now still appear as purple in color because it is intensifying the color of crystal violet. In the third step of gram staining, we will apply a solution known as decolorizer. This decolorizer solution is made up of alcohol and acetone. This decolorizer will be able to remove this crystal violet iodine complex from this gram negative bacteria. This crystalloid iodine complex will be removed by this decolorizer. So bacteria that was purple due to this crystalloid iodine complex is now colorless. Whereas bacteria, gram-positive bacteria having a thick peptidoglycan cell wall, and there is a huge crystalloid iodine complex deposited in the gram-positive cell wall, this decolorizer will not be able to remove this crystalloid iodine complex. So bacteria will still appear as purple in color after this decolorization step. In the fourth step of gram staining, we will apply a stain known as safranine. This safranine will enter the gram-negative bacterial cell wall and color these gram-negative bacteria as pink or red in color. So gram-negative bacteria now will that was colorless due to decolorization, now they will be appear as pink or red in color due to this stain known as safranine. Whereas gram positive bacteria has already crystalloid iodine complex that is deposited in their cell wall, safranine will not be able to enter gram positive bacteria. So gram positive bacteria will still appear as purple in color. This safranine is known as counter stain because it is applied as a second stain. So we call it as a counter stain. Just remember that gram staining is a type of differential staining technique. That means gram staining react differently to different bacterial cell wall and categorize bacteria into two groups: gram positive bacteria and gram negative bacteria on the basis of their cell wall thickness. So we call gram staining a type of differential staining technique that is differentiating between two kind of bacteria. Now we'll talk about the procedure of gram staining. Now we'll talk about the procedure for performing gram staining. In the very first step of gram staining, we'll take a clean glass slide. Then we will put a drop of water in the center of the slide, and then we'll pick a colony with the help of wire loop. Wire loop look like this, right? With the help of wire loop, we'll pick a bacterial colony and mix with the water. And this way we will make our very thin smear, right? We will let this smear air dry. Then we will pass it over the flame 2 to 3 times. That will fix our bacteria onto the surface of slide, so it cannot be washed during the staining procedure. This heat fixation will also kill the bacteria as well. Then we will cover our smear with the stain. Crystal violet, we call it as a primary stain. This was our smear. We will cover this smear with crystal violet, right? We will cover this with crystal violet for 1 minute and let it be on the slide for 1 minute. After 1 minute, we will wash it with clean water. Then we will cover our smear with the help of grams iodine, that was yellowish color solution, right? So we will cover the smear with this grams iodine for 30 seconds to 1 minute. After 30 seconds to 1 minute, we will wash our slide with clean water. Then the very most important step of gram staining is decolorization. For decolorization, we will keep our slide at a 45-degree angle and we will put decolorizing solution drop by drop over the surface of smear. We will apply this uh, decolorizing solution for 5 to 15 seconds, right? So we're keeping our slide at 45-degree angle and we will decolorize for 5 to 15 seconds. This for our smear, drop by drop. This decolorizing solution of ethanol and acetone will decolorize this slide. After 5 to 15 seconds, we will wash our slide with clean water. Decolorization step is very important in a way that we will not decolorize our slide more than 15 seconds because it may decolorize gram positive bacteria as well. So 5 to 15 seconds are enough for decolorization. Then this was our smear. We will cover our smear with another dye. We call it as a counter stain safranin. We will cover our smear with safranin for 1 minute. So after 1 minute, we will wash our slide with clean water and we will let this slide air dry. When our slide is dry, then we will focus it under microscope at first at 10x lens, then 40x lens and 100x lens. Now we will discuss how to interpret the results of gram staining. So after performing the gram staining, we will focus our slide under microscope and there are few possibilities that but how bacteria will look like. So we see round shaped bacteria that are appearing purple in color, right? Round shaped bacteria, purple in color, we call them as gram positive cocci because gram positive bacteria appear as purple in color. If we see rod shaped bacteria that are appear purple in color, we will call such bacteria as gram positive rods. Third possibility could be if we see round shaped bacteria that are appearing pink or red in color. We will call such bacteria as gram negative cocci. Round shaped bacteria are called cocci, right? Fourth possibility could be we see rod shaped bacteria that are appearing pink or red in color. Rod shape and pink in color. We will call such bacteria as gram negative rods. There is another possibility that a bacteria that is not true rods or cocci, but in between rod shape and a cocci, it look like this, for example, right? Such a bacteria is called cocobacilli. Example of cocobacilli is a Haemophilus influenzae. Okay, that's it from this video. If you have any question regarding gram staining, you can ask in the comment section. Thank you. For more microbiology content, please like, share, and subscribe my channel.